Hello students. Today we will talk about endosperm. Endosperm, as we all know, is the most common nutritive tissue for the developing embryos in angiosperms. Among all available nutrition providing tissues, endosperm is the most common. You must be having some idea about how endosperm formation initiates, how endosperm is formed actually. There are two sperm nuclei, one of which fuses with the egg cell or fertilizes the egg cell to give rise to the zygote, while the other one fuses with the polar nuclei to give rise to triploid endosperm. The zygote is diploid and the endosperm is triploid in nature, that is 3N. Okay, the primary endosperm nucleus is normally located directly below the egg cell and undergoes division almost immediately. Please note, almost immediately after its formation. During triple fusion, only the sperm nucleus fuses with the polar nuclei, while the male cytoplasm does not take part in the process. So it is only the nuclei, nucleus which is taking part in the fusion process. The cytoplasm doesn't come from the male side. Okay. Depending upon its mode of development, there are three types of endosperms. One is the nuclear, the other is cellular, the third is helobial. So let's start with nuclear endosperm. Here, the division of the primary endosperm nucleus and a few subsequent nuclear division is not accompanied by wall formation. That means what? The nucleus goes on dividing subsequently without the wall being formed. The wall is not forming immediately after the nucleus is dividing, and as a result, what is happening? After some time, thousands of nuclei remain freely suspended in the central cell of the embryo. Now see, if suppose the structure is like this, if the cell wall is not being formed, what will happen? The nuclei tend to remain suspended like this. Okay. Thousands, few to thousand nuclei, they remain suspended like this. Okay. Such endosperm may persist until it is consumed by the developing embryo. As the embryo is developing, it needs nutrition to grow. Isn't it? It's a very simple concept. When a child is born, it needs ample amount of nutrient to grow. Same thing with the plant embryo. The growing embryo needs nutrition and it consumes nutrition very fast. This type of endosperm persists until it is consumed by the developing embryo, or it may become cellular at a later stage. Initially it is like this. Later it may sometime become cellular. How is it becoming cellular? By wall formation from the periphery towards the center, that is centripetal wall formation takes place. That is, wall formation will start from here towards the center, from the periphery towards the center. Okay. Now this is found in Oxyspora, Limanthus, etc. Fine. See, if you look into the diagram, you'll have a better picture in your head. See, this is your embryo. Fine. This is the primary endosperm nucleus and this is your zygote. You know, this is what? This is 3N and this is what? 2n. These things you know. Endosperm is formed by triple fusion, that is fusion of 3 nuclei. One male gamete fuses with the polar nuclei, giving rise to what? This triploid condition. Now see here in nuclear, what is happening? The embryo is here and the nuclei remain suspended in the cell without being accompanied by wall formation. Okay. Now we'll come to cellular endosperm. We'll now talk about cellular endosperm. In cellular endosperm, from this word you can understand it has got something to do with the wall formation. The nuclear division is accompanied by immediate wall formation. Okay, in cellular endosperm, the division of the primary endosperm nucleus and its further divisions are followed by immediate wall formation. In the previous case, what did we study? The nuclear division was not followed by immediate wall formation. Here it is accompanied by immediate wall formation, and and the three nuclear stage is completely absent. The endosperm is cellular from the early developmental stage. So what do you understand from here? The 3 nuclear stage, this thing is absent. Okay. It becomes cellular right from the early developmental stage. The primary endosperm nucleus divides into 2 daughter nuclei, which is followed by transverse wall formation. Okay, this is commonly found in trimers. Right, now let's take a look into the picture. See, in cellular what is happening? See, the nuclei all have wall around it. All the nuclei here have wall around it. Here see, it is freely suspended. The nuclei are freely suspended here. But here the nuclear division is accompanied by wall formation. Alright. Next we go to Helobial endosperm. This is an intermediate structure between nuclear and cellular types. This is very common in monocots. You can also find this in some dicots, but it is more common in monocots. The primary endosperm nucleus moves to the chalazal end of the embryo sac, where it divides, forming a large micropilar chamber and a small chalazal chamber. So the micropilar chamber is large and the chalazal chamber is small. Keep this in mind. The chalazal endosperm is free nuclear and the micropilar endosperm is free nuclear in the early stage. Later it becomes cellular. Alright. Now see, if we divide endosperm based on the food material, we can have starchy endosperm, oily endosperm and horny endosperm. In starchy endosperm, you see, starch is stored in the form of starch grains, which is commonly found in cereals, in members of OSC. Oily endosperm, oil is stored in endosperm like ricinus, coconut. Endosperm is in horny endosperm is hard due to storage of hemicellulose. You can find it in Borassus. Okay, so this is how we complete our brief and quick revision of endosperm.